A very good evening, all. So, welcome to today's microbiology section. And I'm Avina, and I will be your host. And this class is to deepen your understanding of key concepts and applications in microbiology. So, let's make the section interactive and insightful. So, feel free to ask questions and share your thoughts as we progress. Thank you for joining, and let's get started. I invite Swarnasri ma'am to take over the section.
Thank you, Avina. Hello, everyone. Today, we are going to discuss about the microbiology for post-basic BSc nursing. As you all know, why the microbiology is important for nurses. Anybody? I know everyone know why it is important. Okay, why it is important? Before knowing to importance of microbiology in nursing, first we can know the definition of the microbiology. Microbiology is the study of the biology of microscopic organisms such as virus, bacteria, fungi, algae, or protozoa. Okay, this is the thing, the definition of the microbiology. Or else we can tell like this also. What is microbiology? Micro means a small, okay, or minute, and biology means the study of living organisms, which means we are going to study about the living organisms which will be very small or minute and which we can't able to see in our naked eyes. Understood? So this is the thing, we are going to study about the microorganisms. That is what microbiology. Then we come to know what are the definition of the microbiology. Now the classification of microorganisms. Okay, first one is what? Bacteria, fungi, algae, virus, and protozoa. These are all the classification of the microorganisms which we can able to see in the microbiology. Why we need to study? Being a nurse, what is the importance to study this microbiology? Yes, we are working in a hospital sector. Yes or no? There we are getting more infections. Through which factor we are going to get the infections? What is the main cause? That is what? The microorganisms. Yes or no? So for that reason, we are going to study about the microbiology to curb the infection causing agents like bacteria, virus, and all. Okay, to eradicate those things, we are going to study about this microbiology. It helps nurses to understand and prevent the infection diseases. Okay, both infection and disease. That is the thing, the nurse can able to curb the infections and able to eradicate the disease causing agents. Okay, that is the thing we are going to study the microbiology. The first thing is infection control, then patient care, disease diagnosis. This is very important. Then educating the patient regarding the microorganisms and their causes causing diseases also, then communicating with the healthcare team and developing a care plans. As you all know, in the hospital sector we have one specialized nurse allotted for infection control. You may aware of this, yes or no? For this is the reason we are going to study the microbiology in the nursing field. Now we are diving to the syllabus for post basic BSc nursing. Next slide, ma'am. Next. Yeah, this is the first unit. This is the first unit in microbiology, the structure and classification of the microbes. Already I told you, here we are going to study about the structure of the microorganisms and the classification of microorganisms. When it comes to the structure, we have the questions like 5 marks, that diagram, bacteria diagram. Okay, like that kind of diagrams we are going to see, and different according to their structure only we are going to do the classification. Okay, where the virus will be in the cone shaped or in the spherical shape. For example, staphylococcus and all, it will be in the spherical shape and attached. Like that kind of structure we come to know in the first unit, and their classification like bacteria and the gram positive bacteria, gram negative bacteria, the viruses, algae, fungi. These are all the things we are going to study in the first unit. The next one is morphological types, which means the external feature we will saw, and we are going to do the classification. Types nothing but the classification. Then size and the forms of bacteria. Motility means movement. Then classification of microorganism. That is the thing mainly we are going to cover in this unit. Here, you if you want to write any classification of bacteria or virus, use some flowcharts. Okay, that is the key tip to write in the examination. Here they can ask about the diagrams, and as well as they will ask the classification of microorganisms. Okay, for that reason we should always write in the flowcharts. Then it will be a it will be helpful to get a gain a good marks in the particular question. Okay, next we can move towards the next topic. First unit clear, no? If you have any doubt regarding the first unit, anyone can tell. Hello, am I audible?
Yes, ma'am.
Okay, first unit clear, no?
Yes, clear,
Nothing
ma'am.
is there in this first unit. Okay, there they are going to classify the microbes, which means microorganisms, according to their structure. Okay, according to their structure, this like bacteria, fungi, algae, virus. For example, bacteria means a single celled organisms. Okay, that we are going to classify it into gram positive, gram negative, like this. The next one is fungi. These are all the microorganisms, and mycotic in the sense the fungus. Okay, that is the thing we are going to study. And viruses means which will be in the form of DNA and RNA. It will be non-living organism. Okay, it is a non-living organism. This is the things and their characteristics also we are going to study in this first unit. We can expect the 5 marks questions like classification of microorganism, and the characteristics of bacteria, the structure of bacteria. These are all the things we can cover in this unit. Okay, next slide. Unit second. See, in the unit second, mainly we are going to see the identification of microorganism. Sorry for the inconvenience. In the second unit, we are going to study about the identification of microorganisms and discussion of laboratory methods. Discussion of laboratory methods and diagnosis of bacterial diseases. These are all the things we are going to study in the second unit. Okay, identification of microorganisms, which means whether the disease which will be caused by the bacteria or virus or fungal or algae. This is the thing we are going to identify the microorganisms by studying their structure and all. Okay, in the previous unit we study the structure. No, here we are going to identify the microorganisms. Then the laboratory methods. We have various laboratory methods, which means we are going to collect the various specimens, like we are going to collect the sputum. For what? If we want to check the this is the diagnostic test for tuberculosis, means we will collect the sputum, as well as we will collect the blood to detect the disease. Okay, this is the thing we are going to study in this one. Then diagnosis of bacterial infection. For example, we are going to diagnose the bacterial infections like tuberculosis. We will go through the laboratory test. Okay, that is the things we are going to study in this unit. Okay, we have to deeply know about the laboratory methods, what we are going to do in the laboratory, which means for diagnosis purpose, what and all the test we are going to do in the things. For example, for virus we will do various kind of tests. Okay, that is for bacteria we will do the different kinds of test. Okay, the hematological test, like that kind of things we have to do. Okay, that kind of knowledge we will gain by this unit. The next unit is unit number 3. Unit number 3, here we have growth and nutrition of microbes. Okay, growth and nutrition of microbes, temperature, moisture, blood. In this unit, what we have to do in different temperature. Okay, means whatever the things, living things, it needs some environment, yes or no. For that particular purpose, the growth and development or the growth and nutrition of the microbes needs a particular temperature. For example, it will be in a 60% of temperature, means it can able to grow easily. Okay, then moisture, blood, these are all the things which will need. Okay, some yes sir, if you agree, no, for the microorganisms will be present in our human body to cause the diseases. Okay, that time how it will be present in our blood, it will be in our body, means it will be in our blood. To in that humidity, how it will be grow, how it will get the nutrients. That is the things we are going to study in this unit. You can expect the 5 marks questions regarding the growth and development, or else you can expect the filling the types of questions. That kind of questions we can expect from this unit. The next unit is fourth unit. In this fourth unit, we are going to mainly study about the destruction of the microorganisms. In the fourth unit, the first topic is what? Destruction of the microorganisms. It is very important unit. Here we come to know the methods to destroy the microorganisms. Yes or no? The microorganism can be destroyed physical or chemical methods. Here we are going to study about the sterilization and disinfection. Okay, when it comes to the sterilization and disinfection, there is a difference between the sterilization and disinfection. That is what the sterilization means. The completely 100% we are going to eradicate the microorganisms. But the disinfection means we will eradicate. Okay, on the surface so many microorganisms is there, we are going to clean that by using the disinfectants. But here only the difference is what? We are going to clean 99.9, but in the sterilization we are going to eradicate in 100%. That is what the difference between the sterilization and the disinfection. For example, the sterilization means the process of completely eliminating the microorganism from the surface or the object. If any object or the surface, for example, in the OT and we should always 100% sterilization. Okay, to operate the operation. For that purpose, we need 100% elimination of the microorganisms. During that time and all, we will use the sterilization methods. Okay, then which mean the next one is what? Disinfection. In the disinfection, the process of removing the microorganisms, including potentially pathogenic ones, from the surface or the object, okay, which means we are going to potentially clean the surface or the object to remove the microorganisms, but the sterilization is very much effective than disinfections. Okay, the next one, chemotherapy and the antibiotics, we are going to study in this unit. The effects of heat and cold, again it will be there. The effects of heat and cold means what? Here there is a survival of the microorganisms like bacteria, virus and all. Okay, for that purpose we need particular environment. Sometimes what will happen, for example, we are going to study about the structure and morphology of the microorganisms. There is a growth, then survival, death and adaptation. Sometimes the microorganisms will adapt some things and they will survive in the particular environment. Okay, during in much heat also it can't able to survive. Okay, in much cold temperature also it can't able to survive for some bacteria and all. For that purpose we have to know about the effects of heat and cold. And the next one is what? Hospital infection control procedure and the role of nurse. This is very important. Okay, here we have one specialized area or specialized post for a nurse in the hospital sector, that is infection control nurse. See, we are human beings, yes or no? If we are working in the hospital sector also, we need to protect ourselves. We have some certain protocols and procedures to curb the infection, and we have to protect ourselves, yes or no? For example, during the corona we have to use the ppe, personal protective equipment. Why we are using that kind of things, or sanitizers? Why we are using? Only the thing is we have to
To
protect
protect
ourselves
to protect
from the,
ourselves.
yeah, that is the main thing. That's why we are using that vendor, that and all. And we have to care our staff also in the hospital, yes or no? That is the role of nurse, yes or no? That is the thing, they are having
Yes, ma'am.
infection control nurse in the hospital. Okay, like this we have to know about the infection control in procedures in the hospital. For example, HAI means not high, it's a hospital acquired infection. Yes or no? The hospital is a hub of microorganisms and infections, yes or no? We will used to get
Yes, yes.
infections from the hospital. Some people will tell, no, if any cleanliness which not maintained properly means, if I go this hospital from the here only I will get the infection. In our medical term we will call it as a nosocomial infection, which means the
No
infection which will be
supplement
acquired by the hospital itself.
from the
That is the thing,
hospital.
to curb this one and all, there is a roles and responsibility. She have to take care of those kind of things. She has to maintain properly the cleanliness and all. She has to control and she has to maintain the sterilization methods. These are all the things, the duty of infection control nurse. These are all the things which will be covered by this fourth unit. Any doubt? Here we are going to use some disinfections, then antiseptic solutions, sanitizers. Why we are using the sanitizers? We have to protect ourselves, yes or no? That is the thing. Um, then for cleaning the floor and all, we used to use the phenol. Why? To kill the microorganisms, yes or no? On the surface or object wherever we can able to expect the microorganism presence. Why? We can't able to see in our naked eyes. By the help of microorganism microscope only we can able to see the microorganisms. With our naked eye we can't able to see, right? For that purpose we have to maintain a proper protocol of the infection control in the hospital especially. Otherwise the we have to face the complications, yes or no? Understood?
Yes, ma'am.
Yeah, this is the things we are going to discuss in
Understood.
the fourth unit. Next one is fifth unit. In the fifth unit we are going to discuss about the disease producing microorganisms. So first we can able to take the pathogens. Okay, pathogens nothing but disease producing microorganisms. Understood? Don't be confused in the pathogens. Don't be confused between the pathogens. Okay, pathogens are the disease producing microorganisms. Okay, understood? The next one, first one we can able to take like a tuberculosis. Okay, tuberculosis, it will be caused by the which bacteria? Mycobacterium tuberculosis. Yes or no? Through this bacteria only we will get the disease called tuberculosis. Then leprosy. Leprosy which will be caused by the bacteria called Mycobacterium leprae. Okay, these are the two bacteria which will cause. One is caused by the first one is what? Tuberculosis. The tuberculosis which will be caused by the Mycobacterium tuberculosis. Answer.
It's not clear. Maybe net problem.
It's not clear. Okay, once again I
No,
will
no.
tell. Now it's
Yes,
clear.
please, ma'am.
Okay, once again I will
Yes,
tell.
ma'am.
Unit 5. In this unit 5, we are mainly going to discuss about the disease producing microorganisms. In this disease producing microorganisms means what? Pathogens. Okay. In the pathogens, which will be causes what? Diseases to the human. Yes or
Yes,
no? For
yes, ma'am.
example, we will take tuberculosis and leprosy. Okay. First one is what? Tuberculosis. Am I audible?
Yes, ma'am.
Okay, tuberculosis. In the tuberculosis, this is the disease which will be caused by the Mycobacterium
Microbiology,
tuberculosis. That is the
tuberculosis.
bacteria. That is a causative agent. The next one
Yes,
is what? Leprosy. Leprosy which will be caused by the Mycobacterium leprae. Am I audible?
yes, ma'am.
Okay. The next one is
Microbiology.
anaerobes, cocci, spirochetes, rickettsia. These are all the things we have to learn in this unit. Main thing that is cocci. No. Fifth unit only. Fifth unit, ma'am. Cocci. In this cocci, we have various classification. Okay, let me explain deeply this one. First thing is what? Diplococci. Okay, first one is cocci. Cocci, which means you just imagine cocci means only one. Okay, only one spherical shaped microorganism. Diplococci. Diplococci means what? We have two
Two.
spherical shaped microorganism, means the pair of cocci will be present. Okay, the next one is, the next classification is what? Streptococci. The streptococci means which we can see in the form of rows or chains. Okay, we can able to see in
Yes,
cocci in the form of chain. Okay, or
yes.
rows in the form of row. ROW row. Okay, that is the thing we can see, and we will call it as a streptococci. Don't be confused. Diplococci means what? The pair of cocci. Streptococci
Two.
means what? The rows. Am I audible?
Or chain. Chain, chain.
Yeah. Streptococci means a chain of cocci. The next
Okay.
thing is, the next type is staphylococci. In the staphylococci, we have the structure of the cocci in the form of grape shaped. You can understand, you can imagine that structure. Okay, that is
Yes,
what
ma'am.
called staphylococci. Okay, the next one is Sarcine. Sarcine means what? This is also one of the form of cocci, which will be in the form of packets, or eight or more cocci will be present in the bunch. Okay, understand? Don't be confused.
Yes, ma'am.
Staphylococci means what? The cocci which will be in the form of grapes.
Great shape.
Understanding? Yeah. The next one is Sarcine. Sarcine which will be in the form of packets or in the bunch of cocci. We
Mm.
can expect eight or more cocci will be present. The next one is tetrad. Tetrad, by naming itself, we can able to understand that the tetrad means the four cocci will be present. Understood?
Yes,
These
ma'am.
are all the things we are going to study in the cocci. Okay, cocci, we have discussed that the diplococci. Diplococci means what? The pair of cocci.
Two. Pair of
Streptococci means rows or chain of
chain.
cocci. Staphylococci means grapes
Great
like
shape.
structure, which will be in the form of grapes. Okay, that is what staphylococci.
Okay.
The next one, sarcine. Sarcine is that
Okay.
packet or
Eight.
eight
Eight of
or more cocci will be present in the bunch. The next one is
four.
tetrad. Tetrad, by
Four
naming
perfect.
itself, yeah, by naming itself, we come to know that the tetrad will be having four
Four
cocci.
perfect.
Then here we come to know the gram positive, gram negative. Gram negative means what? The thin peptidoglycan layer will be present. That is what gram negative. Then gram positive
Yeah.
means the thick peptidoglycan layer will be present. Okay. For example, for the cocci is staphylococcus. Okay, these are all the bac- uh, cocci which will cause the
Staphylococcus
diseases.
aureus.
Okay, staphylococcus, yeah, staphylococcus, streptococcus, pneumococcus, neisseria. Okay, and we will call it as
Yes.
staphylococcal infections. Some types are like food poisoning and all, which will be caused by this kind of cocci infections only. Understood? The
Yes.
next one is the spirochetes, the group of bacteria that we can cause the serious diseases to the humans like syphilis and all. Okay, these are all the things which will be the syphilis and all which will be caused by the spirochetes, and which will be a group of bacteria. Spirochetes are the group of bacteria along with the spirochattle. Okay, then their morphology, when it comes to the spirochetes, by naming itself we can able to understand that it will be in the spiral. Okay, which will be in the
Yes.
form of spiral and long, then slender will be there, which will be in the form of spiral shaped bacteria. Okay, then motile. Okay, motile means what? Movement. Okay, rickettsia means what? It's a type of gram-negative bacteria found in lice or flies. Okay, which will be not present in any other human bodies. Okay, which will be in the form of like flies, fleas.
Yes.
Fleas
Yes.
and
Right.
lice will be present. No, we can able to expect the rickettsia. It is a form of negative bacteria. Understood? Now we can move towards the next unit. That is what?
Sorry, ma'am. Flies and what else?
Lice, lice, Lice,
Like.
lice,
Okay.
lice, which will be
Okay.
present in our hair, right?
Hair. Yes.
Usually,
Yes. Yes.
yeah, we can
Nice.
able to see in our hairs. That is what the rickettsia we can able to see in the fleas and lice.
Okay.
Lice. Okay. The next one
Thank
is
you.
unit number 6.
Yes.
Ma'am,
Unit number
excuse
6.
me, ma'am. Excuse
What?
me, ma'am. Tell
Please.
others to tell others to mute the mic because we are not audible properly.
Everyone can mute the mic, no? Please. Please mute your mics. Avina ma'am,
Yes, ma'am. Everyone muted.
you just mute everyone's mic, ma'am. Next, we can move towards the sixth unit. That is what? Pathogenic fungi. Okay. In this pathogenic fungi, here we have to know about one particular terminology. That is what? Mycotic infections. Okay. Mycotic or myco means what? It will tells about the fungus. Okay. Which causes the fungal infections, the pathogenic fungi, which will causes the fungal infections or disease in the humans or other organisms. For example, for the fungus, it is Candida aureus. Fungus Candida aureus or Candida albicans we can able to see in the female, especially regarding this infections. Okay, that is what which will be caused by the Candida albicans. The next one is systemic mycotic infections. When it comes to the systemic mycotic infection, what will happen if something will happen in our body or particular organ which will going to be spread, okay, to entire body. That is what sometimes we can able to see in our skin and all the fungal infections, or it will be able to we can see through entire our body. That is what systemic mycotic infections. Then laboratory diagnosis for the mycotic infections, again we are going to do the skin test and all, like for example blood test. These are all the things which will be hematological test and all, which will be done in the laboratory to diagnose the mycotic infections. Here we come to know if whether they are asking about the mycotic infection or fungal infection. You have to know about that. Only the thing is both are same. Okay, understanding, bro? Understood, right? So don't be confused between the fungal infection and the mycotic infection. You should know about these things. Both are same only. Next unit. Next unit is immunity. Okay, this is very important unit. In this immunity, immunity is the ability to avoid or not to be affected by the disease. Okay, which will be against to the disease condition. Okay, for example, we have active immunity, passive immunity, and these are all the types, and innate immunity. When it comes to the innate immunity, which will be present in our body itself from our birth, and it is going to be lost when the life is going to be lost. Okay, we can able to result in our death, but it will be present by birth. Okay, the body's first line of defense against the harmful substances such as bacteria, virus that are present at birth and last at the person's entire life, which means which will be present in our death, it is going to be lost when the person is going to be died. That is what innate immunity. And we are going to know about the antigen and antibody reaction and immunization. For example, when it comes to the immunization and all, we have to like to secure ourselves from the disease condition, we will take some immunization. Okay, for example, in COVID time we will get immunized by the vaccines, yes or no? In the form of vaccine we taken a immunization to eradicate the disease or to protect our from the disease. Okay, to save our life we will get the immunization. I can't able to discuss the entire immunization schedule in this particular session. Here it will be important, the immunizations schedule, and antigen and antibody reaction. And one more thing is immunity and types of immunity, like active immunity, passive immunity, innate immunity. These are all the things very important. We can move towards the next unit, that is what, eighth unit, the parasites and the vectors. Okay, this is also one of the very important unit, the parasites and the vectors. When it comes to the parasites, which will be not live in any other environment. Okay, it will can't able to live in a different environments, but the vectors are the vehicle or the media that can able to transfer the parasite or parasitic infections to the human being. The parasites are the microorganisms, but the vectors are the vehicles. Okay, for example, vectors, the mosquitoes, fleas, ticks. Okay, these are all the example. See what will happen. We will take one disease condition and we can discuss this unit. It will be very easy to understand. For example, we will take dengue. Okay, dengue, which will be affected by the mosquito. Here what vector will be the mosquito, yes or no? The disease condition which will be transmitted through the vector, it needs one media to transfer the disease. Okay, without the media which can't able to get the disease, the human being will not never get. But if something else which will transfer, okay, that is what vector. Then there are characteristics, classifications of parasites and protozoal infections, including amoebiasis and helminthisis infection. Then diagnosis for each and every disease conditions like parasitic infection. Then vectors and their disease transmitted through the which vector, which means the mosquito will cause the dengue. Okay, then malaria. This is the thing we are going to study in this last unit. This is also one of the very important unit. We can able to expect 10 marks question from this unit. Here, the protozoa, helminthisis, and amoebiasis are the classification of parasites. When it comes to the protozoa, it is a single celled parasite that can infect the blood, brain, and skin, eyes, other parts of the body, they are usually aquatic organisms which will be present in the liquid, which means which can able to live in the blood and all, and that are transmitted through contaminated food, water and fecal matter. Okay, don't be confused. The parasites, there is a classification of the parasites. Under that we are going to study about the protozoa, helminthes and amoebiasis. The protozoa means what? Which will be a single celled parasites that can infect the blood, brain, skin, eyes and other parts of the body. Okay, they are usually aquatic organisms, which means which will be present in the liquid, okay, liquid media. The next one, that are transmitted through the contaminated water or food or fecal matter. The next one is helminthes. Helminth is a general term for parasitic worm that includes flukes, tapeworm, roundworm, and thorny headed worms. Okay, they usually infect the intestinal tract but can also infect the skin, brain, and other tissues. Okay, these are all the things we are going to study in this unit. The next unit is ninth unit, virus. Here also we are going to study about the virus. There are classification of virus and the disease caused by the virus in man and the animal, and their best example is coronavirus. We will take that is the best example, and how it will be developed in the human body and what is the mechanism and how we are going to fight against the virus, and the morphology of the virus according to the classification, how we are going to classify the virus according to the structure based and according to the character. Okay, like some viruses which will be in the form of cone shaped virus we are seeing, no, in the microbiology we will study that one. And the next one is the disease which will be caused by the virus. These are all the things, and how we are going to control the disease, and what and all the protocol to control the disease which will be caused by the virus, and their diagnostic evaluation, how we are going to diagnose the disease condition caused by the virus. That is what the 10th unit. Next we can move towards the sorry ninth unit. The next thing is 10th unit. The 10th unit is microorganisms transmitted through foods. Yes or no? This, yeah, we will get the food poisoning and all. Okay, that is a, there is a different mechanism to get acquire the infection through our food. Okay, there is a, in food also we can see various bacteria. Yes or no? For example, curd we will see the lactobacillus. Yes or no? Understanding?
Yes, yes.
Yeah, same in this unit we are going to study about the microorganisms transmitted through food, but lactobacillus bacilli is the good bacteria. Other bacteria we are going to study, and regarding the food poisoning and the foodborne infections. Okay, the fleas and all which will be get the food poisoning and the foodborne infections. Yes or no? In the, if you are not maintaining the cleanliness during the cooking and all, it will be, if any unhygienic practices we are doing, means what will happen, we will get the foodborne infections through the vectors like fleas and all. Then regarding the food poisoning and all we are going to study in this unit, and we can able to expect the 5 marks question from this unit. Mainly we will get the long essay from the bacteria, then viruses and all. Here it is also one of the small unit, but we have to focus here also. The next thing is what, the 11th unit, biomedical waste management. This is very important unit, and you have to know about the color coding of the biomedical waste management and the maintenance of the biomedical waste management. Otherwise means we have to follow the guidelines which will be present in according to the hospital. Okay, that is the thing we have to learn in this unit. This is also one of the very important unit. Here we can able to expect the questions like 5 marks questions. This is all about the syllabus review of the microbiology. The next one is question paper. This is the previous year question paper. Here first I will explain about the question paper pattern. Okay, that only that much time is there with us. So we have two questions. We have to answer two questions in the first main. Okay, first I will explain the question paper pattern. The first one is we have long essay type of questions, then short essay type of question, short answer. Okay, this is the pattern of the question paper. Here you have to go for 75 marks. You need to attend this examination for the 75 marks. In this question paper pattern, what will happen, we have three questions in the first main itself. We have three questions. We need to choose the only any two questions, which means how much you familiar with your answer. Okay, how much you have confidence on the particular question. That question you have to choose and write. Okay, if you don't have any confidence or you don't know anything about other questions, means you should choose the right one. Okay, otherwise if you and if you delay the time also, it will be very hard to finish the things. Okay, cleverly you have to choose the things. Only two questions we need to answer. Each question carries 10 marks. Here we can able to score 20 marks. Okay, the next thing is short essay. In the short essay we have to answer any 7. Okay, 7 into 5 is equal to 35. Here we are going to carry each questions by 5 marks only. Here we need to attend for only 5 marks. The next one is short answer. We have 10 questions of short answer. Each question carries 2 marks. Here we can able to get the 20 marks. Only one thing we have to focus, means each and every question we should attend. We should not skip any questions, especially 2 marks. You should not tell that 2 marks no, we can easily skip and all. You should not have that kind of mindset. Okay, each and every questions we need to attend. And the meanwhile, what is the thing is there, means certain criteria you have to follow to write the examination. Okay, already I told you the schematic representation and all. But if you have any doubt regarding that, in the last class volar attend my session, means you come to know the schematic representation. Did you get? Hello? Okay, I will share the image. 1 minute.
పోయాడా
Eh? Avina ma'am, I send you one pic. You just share that picture. Hello. See, she will share the pic. Okay. When it comes to this, in the last class also I was unable to attend. It's okay. Now you listen. This is very important part of the session. Okay. When we are writing the examination in last session itself, I told the schematic representation, right? We have to use some like diagrams, okay, the flow charts. If you are going to write the classification, suppose you think like that, you will just imagine, okay, we have to draw two columns, okay. Under that we can write gram positive bacteria, gram negative bacteria. You just highlight those two, okay. Under that you can write, list out the bacteria which will be comes under the gram positive and gram negative, same like that only you have to do. Okay, that is the thing, the schematic representation. Or else you can draw some trees, or else you can draw some flowcharts like flower and all. Okay, not like a flower, but wait 1 minute. Avina ma'am, you just share that one because I am not able to share. Yeah, now you can see, no, here, see, like this only, same like this only, use some different types of schematic representation. You can able to see here, no, you get the idea right now, isn't it? Hello.
Yes, ma'am. Yes,
Anybody?
yes. Thank you.
Yeah. Last time you guys told me I don't know about this schematic representation, right? This is the thing that we can able to use in the examination. Okay, this will be make sure you to get a better marks in the examination. Why I'm telling, keep on telling about this. You are very clever and intelligent. If you don't know how to write the examination, make sure you will fail in that. Okay, that is the same thing you have to use like this one. Not only this one. Okay, you can use some flow charts while writing about the classification and all. We can do from our primary itself. Okay, that is the thing we have to follow in this session, like examination writing pattern. Okay, now you get the idea, right?
Yes, ma'am.
Yeah, this is the very important thing when it comes to this microbiology. You have to know about the culture, the media, okay, the temperature, and which disease which will be caused by this one. Okay, for example, syphilis, gonorrhea, and all, which will be causes the sexually transmitted diseases. Yes or no? Especially the HIV. Okay, you have to know about this both structure of the particular virus and all. Okay, for example, HIV, we have one structure. Yes or no? For each and every virus we have a structure. When we are writing the bacteria also, you have to write the we are going to classify, means the bacteria's types and all. If you want to elaborate the answer, means roughly you draw the diagrams of that. Okay, understood? This is the
Yes,
schematic.
ma'am.
presentation, you should not use much more in every each and every answers, okay? But wherever you need, you know that one, ma'am. You just put this previous year question paper slide. Everyone come to understand this slide, right? Now,
Yes, ma'am.
okay, ma'am. You just put the previous year question paper slide. Okay. See, each and every question we don't need this schematic presentation, but 10 marks for sure we need to use this one. Okay. Then 2 marks also. We can't able to write 1 or 2 pages for 2 marks, right? You have to be calculative in your mind itself while writing the answers. So for that purpose, you have to use the schematic representation. Okay, for example, they will ask about the types of virus, okay, that you use the flow chart, or else some different questions they will ask, okay, that you can simply draw one small tree in this leaf side, no, you just write your answers. Now you get the idea, right?
Yes, ma'am.
Then we are going to write about here they ask the Q fever, the dengue fever. Here we can able to write this signs and symptoms and causes, everything. Now that time we can able to use the schematic presentation. Wherever here itself we come to understand, they mention that your answer should be specified to the questions asked. Draw neat label diagrams whenever necessary. Okay, here they asked in the one question, draw neat label diagram of tropozoa and cyst of Giardia limbra. Okay, this is the thing. If wherever they, whatever the diagram they will ask, you have to write the diagram. Okay, without fail. Means use the black pen only. Some people used to there, they used to use the pencils while drawing the diagrams. Okay, in the university examination, there is no allow of the pencil. Okay, you should always use the black ball pen. It should not be ink pen also, it should be always black ball pen. And you have to draw all the diagrams in the pen only, and you have to mention the parts properly. Okay, then only you will get the entire marks. Okay, the next one, if you want to write about the biomedical waste management, you just, for my sake of knowledge, I used to tell that you just draw three small dustbins. Under that you list out the waste which we are going to keep on this dustbin, okay, which will be very effective to see by the evaluator, and the evaluator will be impressed and they will give full marks. In 2 marks and all, we will get the two, for 2 marks we will get the out of 2 only. Okay, if we are doing using this kind of tricks and all. Okay, for your betterment I'm telling. If you have any doubt regarding this session, you can clarify now itself. Any doubt?
Excuse me,
I'm
ma'am.
not.
For 10 mark questions, how many? Oh, hello.
Hello. Please tell me, ma'am.
Uh, for 10 mark questions and all, how many pages we should be write the answers?
Maximum we need to write 5 to 6 pages. Okay. One thing I will clarify. If they mention paper, can you mute, please?
Yeah, sure.
Okay, if they will mention the number, question number, okay, suppose first question 10 marks question, right? They will give the 10 marks question. First question is what? First number. Okay, first number 5 page means you have to answer only 5 page. Understood? If there is no number which will be present in the answer sheet, means we have to write 5 to 6 or 7 pages for 10 marks. Okay, you can write. Okay, by using the schematic representation, we can elaborate our answer 10 pages also. But if they are given the 5 pages, you can able to write that only 5 pages. Okay,
Okay, ma'am. Thank you.
you just plan dear how to write. Okay, what is the thing? First thing, when it comes to the 10 marks, introduction, then definition of the particular specific point. Then you have to, if you know the author, you just write about the author and according to whom or when. Okay, if you don't know, just left it off. Okay, the next thing is you have to move. Whether they are asking the classification, go with the flow chart. Finish in the next page. Okay, briefly I'm telling about the answer writing because so many people are failing in this particular point itself. Understanding? If you want more information regarding the particular subject, you just go and check out the Nursing Guru app. You will get the entire resource there.
Excuse me, Sonasri ma'am,
Ah, sir.
just 1 minute.
Okay.
Yeah, good evening. Yeah, 1 minute. One more thing. One person has asked that doubt regarding the answer. How many pages we have to write the examination for the essay? Okay.
Yes, sir.
So
Long.
listen, everyone. Okay. Okay. Listen, every student. Okay. In the booklet, each one of the booklet they have the numbers. Okay. So even you are going to write the first essay means you will get the first number page. Okay, the page number will be there, the page number also will be there, and the answer number also will be there on the booklet. Okay, the particular number, the booklet number only you have to write the particular answer. Even you are going to write the 22nd answer, one answer, that question number is in 22 means the 22 question number it will get it in your booklet last page only. Okay, don't write it in the 22 answer in the first page. If you people are written the answers like that, the marks will be cancelled. Okay, so everyone please reminder, that is a main thing. The already the question paper, the question number will be mentioned into that booklet. Okay, the particular page only you have to write the particular answer. Even you are going to write the question number 18. Okay, that question number 18 means you have to go to the booklet and you have to check the booklet. Where is the question number 18 number will be there on the booklet. The particular space only you have to write the 18 questions answers. Okay, the same way because the booklet it will come 1, 2, 3, 4, 5, like that only the question number will be there on the booklet. But you
Yes.
people are going to write the answers because you know the answer of the 2 marks. The 2 marks will be starting from 13. Okay, 1 to 3 is the question paper model means 1 to 3 is a essay, and the 4 to 12 is a 5 marks question, and the 30 to 22 is the 2 marks questions. Okay, like that only the question paper it will come. Okay, so you can, if you have any doubt about the question paper, you can go to the Nursing Guru app. The question paper you will available. Uh, it will available the question paper model. Okay, so the 2 marks will be starting from 13 onwards only, 13 to 22. So you are going to write the answer on 13 means you have to go to the 13th
Page.
question number page. There only you have to write the 13th answer. Okay, so
Yes.
please don't be confused that the many people are facing the same problem. We already told to many students, then also they are going to write the answer in the first page. The mark it will not be evaluated. They will be strike out the paper and they will not be calculated. Then if the answer is right also they will not see that. Okay,
Yes.
everyone
Yes.
understand that.
Yes, sir.
Yes, sir.
That is a main thing. Okay, so that is a main information. So you have to write the particular answer in the particular question paper. Okay,
Okay, sir.
okay. Thank you.
Shall we wind up this session? Thank you.
Any other? Okay, we can wind up. I hope it was an insightful and valuable experiences adding to your knowledge in microbiology. Wishing you all the very best in your studies and future endeavors. Have a great day ahead. Thank you. Thank you so much. Thank you, ma'am.